Thank you for joining this web presentation on TrueORFs, OriGene's 37,000 application-ready cDNA clones for protein expression and functional studies. I'm Carl Kovacs, VP of Customer Services. Here you will learn about OriGene's decade-long effort to clone every single human or mouse gene transcript, as well as the precision shuttle system that provides you with many more experimental options to investigate the function of your gene, starting with a single TrueORF clone. By way of a quick introduction, there are two basic types of clones in the marketplace. OriGene uniquely offers both. The insert of full-length cDNA clones contains the entire transcript, which includes both 5' and 3' untranslated regions, or UTRs, in addition to the protein coding region, or ORF, standing for Open Reading Frame. After in vivo transcription, these full-length transcripts recreate the native and mature mRNA. This can be highly desirable, especially if it is known that the UTRs provide transcript stability or assist in protein translation. OriGene brands these clones as True clones. By contrast, the insert of ORF cDNA clones contain only the protein coding region, but permit the easy tagging of expressed proteins with in-frame fusions to either N-terminal or C-terminal tags. Incidentally, TrueORF clones, as they are known, also have the ability to express only the protein coding region. However, the tags offer a wide variety of highly valuable utilities that we will now discuss in detail. With various tags, ORF clones can be used for multiple applications. OriGene's TrueORF clones all have a CMV promoter capable of driving protein overexpression in mammalian HEC293 T cells, which is seen here at the top left in a Kumasi-stained STS page gel. With a tag, you can detect the protein on a Western blot using our anti-tag antibody or a gene-specific antibody. The Western blot lanes at the top right compare the vector control to the successful gene-specific overexpression. In vivo studies are also possible using fluorescent tags to localize the protein and follow its translocation after IL-6 stimulation, shown at the bottom left. Using organelle-specific proteins, you can easily identify subcellular structures with the aid of different colored fluorescent tags. The challenge to provide genome-wide coverage is great, but OriGene leads the charge by offering the widest coverage of sequence-verified ORF clones that are now expression-tested by Western blot validation. Our ongoing effort provides you with clones in vectors having immediate utility with MYC and DDK, also known as flag tags. The easy shuttling of the ORF insert into over 30 destination vectors increases your access to the leading fusion tag technologies as well. As you will see, a TrueORF clone is application-ready and surprisingly affordable with over 5,000 TrueORF Shuttle clones priced at $380. Let's take a look at why OriGene's premium TrueORF clones are your best choice and why OriGene continually strives to be your gene company. All TrueORF inserts are fully sequenced to represent a unique RefSeq accession number, and you are encouraged to review these on our website to verify their suitability to your needs. By way of comparison to the MGC collection, OriGene uniquely offers over 5,000 so-called tough genes. These include GC-rich as well as large clones. The two charts displayed here indicate the number of clones at each insert length. Please note that OriGene has many more of the larger genes, which are usually in low abundance in standard libraries and therefore are much more difficult to isolate. As a side note, if you need a difficult-to-clone gene that we do not offer from a different species, please take a look at our Exact ORF service accessible on the OriGene website. This will give you access to our custom cloning and gene synthesis expertises. Beyond the sequence validation of a clone, the TrueORF tagging in the entry vector permits the ultimate validation, protein expression. Here we show the vector control and vinculin TrueORF clone expression after transfection into 293 T cells. The cell lysates were analyzed by Western blot using an anti-tag antibody that confirms the 3.2 kb ORF overexpression at the correct protein size, 110 kilodaltons. No other clone vendors are able to offer you this level of assurance. The Western data is available online with currently over 5,000 TrueORFs having already been validated. This ongoing validation effort will increase the number to over 10,000 by the end of Q2 of 2009. This will continue until completed. Here you see several validation examples as Western blots of proteins ranging in size from 11 kilodaltons for APO3C to the very large protein LRP1, which is 504 kilodaltons. OriGene gave much thought in designing the vector to house the valuable TrueORF collection. PCMV6 entry is a fully functional vector for protein expression, rather than a mere archival vector. It has the CMV promoter to drive mammalian cells expression. It has the MYC and DDK fusion tags for purification and identification. It has the neomycin selection marker to establish stable cell lines. And it has the T7 promoter to drive protein expression in a cell-free system. OriGene's entry vector with all these functions is consistently used within the entire ORF collection. This is something you will not find with the entry vectors from other shuttling systems. Precision Shuttle is the name of OriGene's shuttling system designed to move your ORF insert into other destination vectors having new functionalities. This is made simple with the use of 2 rare restriction sites coupled with the use of different E. coli antibiotic resistance selection markers between the entry and the destination vectors. The list of destination vector options continues to expand and is unlimited, but currently contains tags on either the N-terminus or C-terminus, those being fluorescent protein tags for cellular localization, or HIS, HA, GST, DDK, flag, or MYC for protein purification and identification. The latest E. coli expression vectors, inducible vectors, and alternative mammalian selection markers are also available. The simple cut-and-ligate procedure for shuttling in Precision Shuttle system is highlighted by the use of the SGF1 and MLU1 sites, which do not cut within over 96% of all human ORFs. This diagram illustrates the parallel digestions of an entry vector TrueORF and your destination vector of choice. After a simple dephosphorylation of the empty target vector, simply mix the 2 digest reactions, ligate, and screen for ampicillin-resistant colonies. Note that the undesired entry vector is kanamycin-resistant and will not produce any colonies. Also note that every destination vector is already engineered to produce the proper in-frame fusion, and this whole process takes only 2 hours. Let's compare this now to the gateway shuttling system. 1. The entry vector can be used for tagged expression in mammalian cells and in cell-free systems. For many applications, there is no need to shuttle the ORF to a destination vector. In the gateway system, the entry vector is only a harboring place for the ORF. Only when the insert is recombined with the destination vector does the plasmid become functional. 2. The precision shuttling is accomplished with a simple restriction enzyme digestion, then ligation, without the need for an expensive recombination-specific enzyme. 3. There are no intellectual property restrictions imposed by OriGene for any users. 4. Large ORFs up to 18 kilobase can be readily transferred using the Precision Shuttle system, while ORFs larger than 4 kb are unstable in the gateway recombination-based systems. Lastly, the Precision Shuttle vectors precisely add the intended tag to its desired location. There is no appendage of multiple amino acids on both ends of the protein. The utility of TrueORF is greatly enhanced with a large panel of destination vectors available for shuttling open reading frames from the TrueORF entry vectors. These can be classified by both their expression platform and their downstream utility. A few popular options are shown here, and we will show some examples of these applications in the next few slides. Our website lists our expanding set of destination vectors, be it epitope tagging for detection and purification, fluorescent protein tagging for subcellular localization, drug selection markers for establishing stable cell lines, inducible expression, or mammalian and E. coli protein expression. Here we show the results after a first-step purification with anti-DDK beads, a resin to recognize the DDK tag fused to the protein produced by TrueORF clones. These are proteins produced in mammalian cells, 293T, harvested 72 hours post-transfection, with the TrueORFs listed across the top of the figure. It is impressive to see the amount of overexpressed protein from mammalian cell culture, so much that it can even be seen on low-sensitivity Kumasi-stained SDS page gels. For further purification, the second epitope tag MIC can be used in a second step, as well as size exclusion chromatography. There are many citations that illustrate the utility of TrueORF clones. In this Nature paper, the authors expressed TrueORF clones and purified the tagged proteins using the C-terminal MIC tag. These proteins retained their biological function, and they discovered the active resolvase enzymes that regulate the genetic recombination events that occur at holiday junctions. The utility of fluorescent protein fusions can be used to identify subcellular structures. OriGene TrueORFs for proteins specific for each organelle were expressed and visualized inside mammalian cells. The exact protein used is indicated within parentheses, and each was successfully able to effectively paint the endosomes, lysosomes, nuclei, cytoskeleton, autophagosome, as well as focal adhesion. In these cases, we used either green, red, or yellow fluorescent protein tags. The upper panel shows a cell transfected with the fluorescently labeled actin TrueORF clone. Note the filopodia and stress fiber formations. The lower image shows STAT3 tagged with N-terminal GFP in panel A. The green tagged protein translocates from cytoplasm into the nucleus upon IL-6 treatment, and this is shown in panel B. To facilitate protein production in E. coli, OriGene created several destination vectors for this purpose. These contain options for GST tagging as well as tag cleavage by the TEV protease. The left image shows the successful induction of protein expression after the addition of IPTG. In these cases, the expressed protein is fused to a GST protein which was used in its purification. The right image shows the purified fusion protein at 0 hours and then at 3 and 6 hours after TEV protease treatment. The presence of the TEV recognition sequence between the desired protein and the GST tag permits the quantitative cleavage and separation of the tag after purification. Please remember that you can switch from the mammalian cell protein expression studies using your TrueORF to this E. coli expression option through the simple shuttling process made possible by the Precision Shuttle system. When you purchase a TrueORF clone, you can expect to receive highly purified DNA from single-use ion exchange columns. These stocks are confirmed to be extremely low in endotoxin content, and every lot is resequenced on the 5' end to reconfirm its identity after purification. With the 10 to 20 micrograms of DNA that is delivered, you can start your experiment immediately because you don't need to go through the first 3 days of purification and verification that is required when you receive a bacterial stock. Please let us know if you require larger quantities of the DNA. We have quite a large capacity within our manufacturing facility. Let's summarize the TrueORF advantages from OriGene. 1. The clones and stocks have the best in industry QC standards with sequence guarantees and Western validations. OriGene has the broadest genome-wide coverage so you can find what you need. 2. The Precision Shuttle system opens the door to many diverse application options for a large and expanding list of destination vectors. 3. You will receive highly purified plasmid DNA as your starting material, saving days of effort, allowing you to begin your experiment immediately. And lastly, number 4, over 10,000 such TrueORF clones are now available for next-day delivery, with that number expanding rapidly. For more information, I invite you to read more on our website at www.origene.com/ORF, ORF, or contact our technical support team for personal assistance. OriGene is a gene-centric company developing product solutions for gene function research. Our products now cover DNA research, RNAi research, gene expression analysis, and now proteins and antibodies as well. Our product offerings will significantly increase over the next 6 to 12 months, and I encourage everyone to visit our website often. We would love to hear from you about your specific needs and how we can serve you better. Please visit us at www.origene.com or give us a call at 340-3188. Thank you for your time.